Welcome to Inert Chemistry. In today's video lecture, we discuss a very important topic, and our topic is metabolic defect of branched chain amino acids. This is a very important long question related to your past papers. So let's discuss it. The metabolic defect that is related to branched chain amino acid is called maple serum urine disease. It is a rare genetic disorder, and during this disorder, our body fails to break down the branched chain amino acids like leucine, isoleucine, and valine. In this disease, maple serum urine disease, our body is not able to metabolize the leucine, isoleucine, and valine properly. So this leads to buildup of these amino acids and their toxic byproducts in our body. Because our body is not able to metabolize them properly, so they are accumulated in our body. And they can cause various symptoms like sweet odor in urine, also called neurological problems, developmental delays, and intellectual disabilities. The treatment involves the strict low protein diet and medical supervision to manage the branched chain amino acid levels. What is the biochemistry of this disease? Let's discuss in next. In order to understand the maple serum urine disease properly, first we see briefly about the metabolism of branched chain amino acids. There are three branched chain amino acids that here you see leucine, valine, and isoleucine. During the metabolism of these branched chain amino acids, the first step is transamination. This step is catalyzed by an enzyme, branched chain amino acid aminotransferase. And as you know that during the transamination, the transfer of amino group from an amino acid to keto acid takes place. So the same here occurs from leucine. Alpha keto isocaproic acid is formed. From valine, alpha keto valeric acid is formed, and from isoleucine, alpha keto beta methyl valeric acid is formed. So during the first step of transamination, these alpha keto acids are formed. And the second step is oxidative decarboxylation. All these alpha keto acids that are formed, they undergo the second step, that is oxidative decarboxylation. And from alpha keto isocaproic acid, isovaleryl CoA is formed. From alpha keto isovaleric acid, isobutyryl CoA is formed. And from alpha keto beta methyl valeric acid, alpha methyl butyryl CoA is formed. This second step of oxidative decarboxylation is catalyzed by an enzyme, that is branched chain alpha keto acid dehydrogenase. This enzyme, branched chain alpha keto acid dehydrogenase, it is a very complex enzyme and it requires various cofactors for its working, like thiamine pyrophosphate, lipoic acid, FAD, NAD+, and coenzyme A. During the maple serum urine disease, this enzyme, branched chain alpha keto acid dehydrogenase, is not working properly. So these alpha keto acids are then further do not processed, and they are not further, and the further reaction do not proceed. So here you see that oxidative decarboxylation of branched chain amino acids is deficient in maple serum urine disease. This is again an overview of the metabolism of branched chain amino acids. Now we discuss maple serum urine disease in somewhat bit detail. As I have explained in the previous video that during the maple serum urine disease, the enzyme branched chain alpha keto acid dehydrogenase is not working properly, and therefore the alpha keto acids are not processed further and they are accumulated. This maple serum urine disease is an autosomal recessive disease in which there is a partial or complete deficiency of branched chain alpha keto acid dehydrogenase. This branched chain alpha keto dehydrogenase is an enzyme that decarboxylates the leucine, isoleucine, and valine. This disease leads to the accumulation of these amino acids and branched chain alpha keto acids, that are these three. What are the characteristics of these maple serum urine disease? It has following different characteristics. The patient show feeding problems, vomiting, dehydration, severe metabolic acidosis, and classic maple serum odor to the urine. If we talk about the treatment, then given a synthetic formula that contains limited amount of leucine, isoleucine, and valine. Here again we have added this slide so that it is very helpful for you to write this question as a short and long answer in examination. You can read it this slide carefully so that you may able to write this in the exam. Thanks for watching this video. Please like the video, share and subscribe the channel.